I started by drafting 8 questions, so feel free to pause this video and you can take a look at the 8 questions. You can do a print screen and try to answer them. And then I will be answering them as we go along, and you will see how correct or incorrect you may have been and understand better the concept by understanding the answers to these questions. Let's review some examples from the last 50 years of how long has it taken to develop a vaccine. In the red vertical line at the beginning of the blue bar, we are going to see the date when the disease was identified, and the gray line at the end is going to identify when the vaccine was licensed. So when we look at the first example on Zika, even though the disease was identified in 1947 and there's been development of a potential vaccine, it is still ongoing. In the case of chickenpox, please note that there's a star because this is the date when the source of the disease was identified. This is probably because chickenpox has been probably described since millennia before our common era, and this would have made the bars completely microscopic if we actually see when chickenpox was first identified. So in these cases, when the source of the disease was identified, and from 1953, it took until 1995 to have a vaccine license. In this other case, hepatitis, same thing, when the source was identified from 1965 to 1981. As you can see, it gets better and better. These bars are being reduced, and now we're going down to about 16 years for the successful vaccine license. However, when we start looking at more complex diseases like Ebola, described in 1976, vaccine actually licensed in 2019, but for the AIDS virus is still ongoing. And then for the SARS virus, it was canceled. And MERS, which is a variation of another SARS virus, which is the Middle Eastern Respiratory Syndrome virus, is still ongoing. The source is from the World Health Organization, and I'm sure that there's a lot more examples that you can look for, but basically generating a vaccine, even when the pathogen or the source of the disease may be identified, is not easy, is not fast, and is not necessarily a straightforward process. Using this publication from the Scientific American in June 2020, I will walk you through the steps for developing a virus vaccine. And basically, the idea of a vaccination is to promote the immune system to mount a response that will be protective. Now, the vaccine itself needs to be a safe version of the disease-causing virus. And this is the key difference between a vaccination and an inoculation. For more details, please go to my other YouTube video, which is called Origins, the First Vaccine. When we look at the variola virus, which is responsible for smallpox disease, we actually see that it is very close to the cowpox virus. If you look at the virus classifications, they really are very similar. It's just that the smallpox, the variola virus, causes smallpox, which is a disease that decimated populations throughout the history of humankind. Whereas cowpox is a disease that affects cattle. Now, the observation in the late 18th century was that people that had been exposed to cowpox, they did develop some symptoms, but nothing similar to smallpox. But most importantly, they developed defenses that now also protected them against acquiring smallpox. These individuals had never been exposed to smallpox and were not ever infected with smallpox because they had created defenses in the immune system. That's what we call cross-reactivity. The defenses against the cowpox, which is the one they had been exposed to, were cross-reacting with smallpox. And this led to the birth of the vaccine field and vaccinations, because now using cowpox was a safe version of a disease-causing virus, but it will protect against the worst disease. So when we look at the cartoon in Scientific American, what they explain is basically, well, what we need to do is, short of finding another one of these situations like in the cowpox and variola virus difference, if we don't have that luxury of having already in nature a virus that's very close to the one causing disease in humans, but it's safer, then the alternative that we have is the whole field of vaccine development is to look at this virus and adapt it for a vaccine. When a virus is adapted for a vaccine, it creates a safer version, but it still has to have the elements necessary for the immune system to identify the original virus. And also it can remember, so immune system can remember how to fight the original disease. So let's walk through this cartoon by Scientific American and see what are the different ways that we have to adapt a virus successfully for a vaccine. First example was when we attenuate the virus. So attenuating the virus is usually done by having the virus in several different cycles or passes through different tissues. And these could be of different animal origin. So basically a virus needs a host cell to replicate. The virus cannot replicate if it doesn't invade or infect a host cell. And the viruses can infect different kinds of cells, sometimes is limited to different kinds of animals or species. And by passing the virus in culture, so this is a biological method, using different kinds of tissue culture, it can attenuate the virus in a way that it weakens the genetic material of the virus while preserving the elements that the immune system will recognize and will cross-react with the original non-attenuated virus. Examples of this vaccine go back to the Kaprowski and Sabin oral polio vaccine, and they are still in use, like for instance in the measles, mumps, and rubella vaccination. The next in the cartoon is inactivating a virus. So what this does is basically inactivate it by using a chemical process. So after using the biological process of passing it and culturing it in biological tissue, there's usually a step that requires something like formaldehyde. So it's a chemical compound that will actually completely inactivate or degrade the virus. This was used in the Salk injected polio vaccine and is still in use, for instance, for hepatitis A virus. Both methodologies are adapting the whole virus for a vaccine in a way that it's safer, but it still preserves the elements that our immune system will use to fight the original disease. And they have different ways of doing it, and there's pros and cons. The third way of adapting is now using isolated pieces or subunits of the virus. So in this example with hepatitis B virus, it's a very complex structure, and we have the hepatitis B surface antigen. Now, this particular cartoon that is taken from Wikipedia is showing the virus as a circle, but it's really not a circle. It's a sphere. It's like a ball. So basically what the virus is all around it. We only see like the purple donuts, the surface antigen in the ball. Everything else is inside the virus. So when a vaccine is just using the surface antigen for the vaccine, the immune system is still seeing the ball as the ball that would be the virus. However, the vaccine is very safe because it does not contain any other element of the viral particle to infect. So it basically has the same structure for the immune system. What the immune system sees is this ball, this little virus. It looks exactly the same. the vaccine and the original virus, but the vaccine does not have any other component that could infect the individual. And this is a technology used for hepatitis B vaccination. It is one of the technologies that has been currently used for the coronavirus vaccine approach, where the spike protein in the coronavirus vaccine is the one that the virus is using to penetrate into the host cell. So by using the spike protein to mount an immune response, that immune response will block the piece of the virus that is used to infect cells. Some other technologies using the same approach would go on to even using just the receptor binding site protein, which is within the spike protein, but it's a smaller one so that you can have more specific immune response to it. Let's take a look at the questions as true or false statements. Number 1, vaccination is the principle used for millennia to expose people to controlled doses of the pathogenic virus to generate immunity. This is false because it's not the pathogenic virus, the material that is used in vaccination. For more details, please look my other video in YouTube called Origins, the First Vaccine. Number 2, vaccines have required an average of 20 years from the first identification of the disease to being available in the market. This is not true. It has taken a lot longer than that. I just gave an example in a table from the World Health Organization, but you can Google it and find out it's a lot longer than 20 years. Vaccines can use safer materials by adapting the pathogenic virus to mount a defensive immune response. This is true. This is the principle of vaccination. The biological methodology is the best way to adapt a pathogenic virus, creating a safer viral material for a vaccine. This is not true. It's one way, and each of the different methodologies that we have currently available for adapting this material can be used, and each one has to be tailored to the virus, to the cycle of the virus, to the way that it responds, and to the way that we can still preserve, after adapting it to a safer version, that it can still preserve the immunogenic capacity. In other words, that can still make the patients injected mount an immune response to the original virus. So we have to take a lot of things in account to choose which methodology is the best for each one of the vaccines. It's a tailored process. Modified whole virus is no longer used to generate material for vaccines. This is not true. We still have modified whole virus, which could be either attenuated or inactivated in vaccines used very successfully today. And number 6, using subunits of the pathogenic virus that are immunogenic is a safe and effective way to generate material for a vaccine. This is true, and the explanation I gave was using the hepatitis B example. Let's move on. We move now to the nucleic acid technology for creating a vaccine. This requires to map the virus genetic sequence and use it, engineering it in a different blueprint. There's 3 blueprints explained here in the Scientific American paper. Basically, the first blueprint is using a piece of the DNA or a specific gene and embedded in a DNA plasmid. The other one is actually using the same piece of that gene that we're interested in and put it in a membrane lipid or a nanoparticle. And the third one is using an adenovirus where we have genetically engineered this piece of gene. So the whole idea is to choose a gene that will encode a piece of the virus that is the one that will create the cross-reactivity to the whole virus. So a little bit of the idea that we saw before of using an isolated piece of the virus or a subunit, that is the best unit for creating an immune response. In this case, we are actually using the gene from the virus that encodes for that unit. Viruses are not living cells. Virus needs a host cell to replicate. They do not have all the machinery. They use the host cell machinery to create more proteins that compose the virus. And they contain within their viral particles all the genes that are unique to the virus and necessary to create more viral particles. That's the minimum amount of information that they need to create it. So if we have a DNA, in this case, the non-coding strand, I put it in green, and I put in red the coding strand. In other words, in the DNA, you have one strand that contains all the genes. The other strand is basically the complementary sequences that protect the coding strand. An RNA polymerase will create the message RNA that is complementary to the gene that it's reading. And in this case, because I'm showing here a DNA with the genes, let's say this is a viral DNA, it would be a DNA-dependent RNA polymerase. And again, this machinery is now going to be using the host cell. The virus cannot replicate itself. So once it gets into a host cell, its genetic pieces are embedded within our own cells. They infect us, they put their genes in our system, and our system is fooled, reading this DNA. Now we're going to be producing an RNA that's complementary to the gene, usually starting with an adenine, uracil, guanine sequence, and ending with any one of 3 potential stop sequences. This messenger RNA contains now the genetic information of the virus. And we take it into our cells, we produce the messenger RNA, adding the polyadenine tail that will identify our ribosomes and our machinery inside to generate a protein from this gene. Our cells are fooled. We think we are actually producing a gene that's ours because the virus is very tricky and it introduces its genetic material into our own infected cells. So we start producing proteins, and there's going to be one gene per protein needed for the virus. So in this case, the example I'm using is hepatitis B that has many different proteins. So there's going to be one gene for each one of these, and we will be producing all of these proteins in the infected cell, and we actually assemble within our cells this virus, and it just continues to replicate and continues to be shed outside our cells and expand. So this is the way that a DNA virus will actually be producing itself, its replication. By using the technology, if it's a DNA virus, the technology of nucleic acid for vaccination, we could also think about using just the gene that encodes for the surface antigen, for instance, and then our body will be producing the surface antigen. So instead of having a vaccine that is injecting the surface antigen protein, we are using a vaccine that has the gene for the surface antigen, and our body will be producing that particular protein so that our immune system responds to it. In an RNA virus, we basically have only one coding strand, and it's the RNA strand that contains all the genetic information. The RNA-dependent RNA polymerase will use our own system to produce the message RNA, which is complementary to the genetic sequence. It has the same start and end codons as I showed before for DNA, and then our cells that are infected will produce the messenger RNA that's adding the poly A tail. This is what allows our own mechanism, our own cells that are infected, to start synthesizing the proteins that are they are actually the ones from the virus. When we are using a particular gene for a vaccine, in this case, let's use the example of the SARS-CoV-2, what we would be using, for instance, is a gene that could be encoding for the spike protein. So our own body will now be encoding that spike protein. And the difference with the previous methodology of using just the subunit or just using the protein from the virus is that we are going to be using the genetic material in the vaccine, and our body will be producing the protein that is the viral protein. Now, what's important is that for this technology using virus genes, or genes from the virus as a vaccine, is that our body will then be producing the viral protein, and our body will be reacting to it using the immune system for reactivity. With this technology, we basically have been using the coding strand, either a DNA or an RNA from the virus. And that's what we have been so far using for vaccination. A more recent technology has been developed and is under development also for some COVID vaccines, which is using the messenger RNA. So the difference between using the RNA from the virus or a messenger RNA is that the messenger RNA has, remember, the complementary sequence to the coding strand, but it's one step closer to making our system produce the actual protein that we need. So it's one less step for our body to produce the protein that will be eliciting the immune response that we need for a vaccine. So you will actually see that in the current landscape, there's both technologies for the nucleic acid, both using the RNA and messenger RNA are included in the current technologies being pursued for COVID-19 disease. So let's review the last two questions as being true or false statements. Number 7, nucleic acid technology to generate the material for a vaccine can be used for both DNA virus or RNA virus diseases. This is true. We can use the genes that are encoded by the virus either in a DNA molecule or an RNA molecule, and this has been used successfully in vaccines. The messenger RNA encoding for the immunogenic viral material is a method used by many vaccines in the market in 2020 and currently one of the strategies for COVID-19 vaccine. It was a tricky question because it is not true that there's vaccines in the market that use this technology yet. There's some vaccines that are in development using this technology, and it is true that this is currently one of the strategies that is being used for COVID-19 vaccines. So the statement as a whole is false because they're not in the market yet. Please check my YouTube channel. If you like it, subscribe. And I do have several other talks, especially on the immune system, immunogenicity, and more specifically on the SARS-CoV-2 and COVID-19. Thank you very much.